thank you very much for this amazing award to the Birnstiel Foundation and the IMP for inviting us all over this today. I will talk about how we can use AI to understand more how regulatory genomics works. And I'm really happy that my talk was after the previous one, because now I kind of can just briefly mention that these enhancers are very important for understanding very complex diseases and also generally tissue differentiation, etc. Enhancers together with silencers are these dysregulatory elements or CREs that are in the non-coding genome, and they can be really kilobases away from their target gene, and they up or down regulate the gene expression. And so at the Cool Lab, we're very interested in understanding where these dysregulatory elements are, how they come together to determine the exact gene expression levels from any given gene. These are kind of hard to study, as was mentioned before already, but there are some amazing techniques for this. The first is performing functional genomics assays, where we essentially genome-wide can track the functional activity through ATAC-seq or similar assays. And these are high throughput and relatively cheap to run, but they only provide a descriptive understanding of the state of the genome. And although they can be indicative of enhancer activity, as was mentioned previously, they don't really provide a model for how different enhancers come together to act on a given target gene. And on the other hand, we also have perturbation experiments, which was also alluded to before. For example, CRISPR interference, where we can directly change the genomic sequence or the regulatory elements, enhancers, by inactivating them. And then we can track how gene expression changes across the genome to identify the target genes. And this provides very nice causal understanding of where these enhancers are, but they are lower throughput and very costly to run as well. And so they are usually targeted at, for example, open chromatin regions only of the genome. And to overcome these kind of limitations, our lab uses artificial intelligence or deep learning models. As you have probably heard about this from everyday life already, ChatGPT or DALL-E 2 have gained impressive performance in various different tasks. At our lab, have been trying to kind of utilize these kinds of models for genomics as well. And to give a bit more detail on the specifics of how we can go from ChatGPT-like models to models of the genome, I wanted to just give a very simplified example of how these models are trained. So deep neural networks, or DNNs, are these black box models that can approximate any kind of a complex function when they are given enough inputs and enough labels for those inputs. So this can really range from any kind of a task, for example, image classification, or in our case, of course, we are interested in understanding the DNA. So the inputs can be DNA sequences, and the output can be some sort of a readout of the chromatin state. For example, is the chromatin open, which we can obtain through ATAC-seq, for example. As you can see here in this large black square, we usually refer to these models as black box because they are highly over-parameterized, meaning that they have a lot of parameters, which makes them very flexible to model these complex interactions within the genome. But that also renders them kind of not transparent, so we can't really learn directly from the model why it's making a certain prediction. And this is something that we'll tackle later on. So just to give a brief overview of my thesis project, the first half was on this package that we released called Gopher, which was a collaboration work with Amber Tang in the lab. And here we were trying to kind of reduce the computational costs of each lab, trying to perform the same tasks of data processing, model training, and evaluation in a kind of unified package. And we also put a lot of thought in how to kind of compare different models that are trained, of course, across different platforms. But in the interest of time, I will not talk about this first project today, and I will focus more on CREM, which is the second package that we worked on, which is on interpretability of these large-scale models. So to motivate this project a bit more, or specifically, I have to kind of step back a bit and tell that the field is very interested in predicting expression levels also, in order to kind of map these enhancer regions in the genome. And in order to do this efficiently, the trend in the field of predicting gene expression is to incorporate larger or longer DNA sequences as input. So we really saw within the last 10 years, expanding from hundreds of base pairs to Informer, for example, predicting gene expression through cage track prediction for a 200,000 base pair DNA sequence. I will not show this in detail, but this is a publication that you can, of course, refer and check this, that the performance of this model is really impressive across the genome. However, as I mentioned before, this is a black box model, meaning that we can't really look at the model and tell why it's making a certain prediction. However, to understand this a bit more in detail, the authors performed in silico mutagenesis, for example, where the individual nucleotides can be perturbed and their importance can be mapped to nucleotide resolution detail, in this case, for example, implicating an SP1 binding site for a SNP or a complex genetic disease. And so this is really nice, but this kind of analysis is usually limited to a shorter input sequence, for example, 200 base pairs. And this really doesn't give a concrete reason why Informer makes a given prediction of high or low gene expression levels from a given gene. And the second method that the authors used was validation using experimental data. And in this case, the authors kind of replicated CRISPR-I experiments in silico in order to understand if Informer agrees with experimental data of where enhancer and gene pairs are. You can see, I will not go into details of how they actually did this, but through looking at the attention maps of the model, that the authors found nice correspondence between the experimental data and the data from the model. In this case, this is, again, very valuable as a methodology, but this is limited to the availability of experimental data and doesn't really tell us anything about what the model has learned beyond the known biology. And to overcome these limitations, we developed CREM at the QLab, which is a new interpretability method for performing large-scale in silico perturbations to understand where these enhancers and silencers are within target genes. And importantly, we can, because we're not limited by experimental costs, etc., we could do this across many genes at the same time to kind of reveal general patterns in gene expression regulation. And at the very high level, the way this worked was that we had sequences of interest that were centered at the TSS with high predicted activity, and then we could do systematic evaluation of how important specific large tiles of input sequence are for the model predictions. And we used CAGE as a proxy for gene expression and focused on K562 cell lines specifically. And again, in the interest of time, I will not go through every single perturbation experiment that we performed, which are detailed in the center of this slide, but I will just give a flavor of a couple of them to just show what can be done in this toolkit. So the first one is what we call a necessity test, where we ask kind of the opposite question of the classical way in which enhancers are defined by sufficiency. And now we ask whether a certain tile is necessary for TSS activation, very similar to how CRISPR inactivation experiments are done. So in this case, we have an example gene that is centered at an active TSS site. And instead of going and specifically targeting the regions for in silico perturbation that are known enhancers from experiments, we now go and systematically split the entire context of the gene into 5 kilobase tiles, and then shuffle each individual tile in order to kind of disrupt any TF binding motifs or regulatory activity that the tile might have. And then we ask, how much does that affect the predictions from the model? What we can see across different classes of sequences with different regulatory landscapes that we call enhancing, neutral, and silencing, we can see a range of different normalized shuffle effect sizes, meaning that there are enhancing and silencing tiles across these genes. Right, so this is one way to kind of define the enhancers in a model-derived manner rather than relying on experimental data. And an additional question that we asked on top of this was to see if there are any higher-order interactions between these regulatory elements. So if we assume that everything is independent of each other in the regulatory landscapes, then we could just perform that one experiment and call it a day. But there is a chance that there are higher-order interactions between enhancers where perturbing one is going to change the behavior of the other enhancer. And that's what we're interested in testing here. So we had a very similar setup to before, but now after we performed the first iteration for searching for an enhancing tile, we kept that one shuffled and then went on to do a second set of iterations where we had already kind of removed the effect of that first enhancer in order to understand if there are any redundant enhancers, for example, whose role was kind of overshadowed by this stronger enhancer in the genetic context. And so we can track these kinds of perturbations across the iterations of shuffling, if that makes sense, on this study diagram. And when we look at the real kind of averaged results across different populations of sequences, we can see that we have around 4 to 5 enhancing Cres on average within the enhancing landscape genes that reduce the activity of TSS is greater than 80%, which is quite interesting. So what this means is that every single gene, according to Informer, of course, has more than one regulatory element. And the implications of this are that if we want to kind of fully describe the system, the regulatory landscape of a gene, we might need to run experiments where multiple elements are perturbed at the same time. Similarly, we ran experiments for silencers, and that did not render that well as well. But basically, we found very similar results also for silencers, where there are groups of these elements within the regulatory landscape of a gene. And to conclude, we have shown that Informer learns a complex set of regulatory interactions, and this can be used to characterize the cis-regulatory landscape of a gene. But of course, this is entirely kind of done through the lens of the model. And we, of course, don't claim that this is the actual real biology, but what we hope to achieve through this is that we can guide future experimental design to kind of narrow down the search space for interesting elements that will, of course, then need to be validated in real life. And then finally, if coupled with other techniques, this can also hopefully facilitate the search for new therapeutic targets. And with that, I would like to thank Cold Spring Harbor Laboratory and the School of Biological Sciences for nominating me, and of course the Koo Lab and Peter Koo for his guidance throughout my PhD. And you for your attention. Thank you.